You know, and I saw this in some of the trial results on winter kill. That's a trait that you get maybe once a decade that you get selection for, and we got it this year. Boy, all the I apologize for the weeds, but that's the result of the winter kill there, you know, and the spottiness. Now, one line that I saved the best one for last. You know, this is one line that I'm fairly excited about. But you can't get too excited. We've been testing this line for 3 years now, and it has exceptional levels of scab resistance. Very, very, very low VOM levels, very low infection index in the field. Its FHB index is very, very low. That means when scab lands on this plant, there's only going to be infection on that one spikelet. Out of 16 to 20 spikelets on there, it's only going to be that one. Just stop scab right in its tracks. And this is the result of decades of breeding and selection. The first cross was made with a Chinese wheat from boy, I forget the region. W14 is the name, Wuhan 14. This was not adapted to Michigan, not a Michigan white wheat, but after years of selection, this line now is 75% ambassador. So you can think of this line as ambassador with exceptional scab resistance. It's got a major gene, so there was a lot of technology that was implemented in the development of this variety. In the breeding process, there was DNA taken from the tissue, and we used a process called marker-assisted selection. So there's a gene on chromosome 3B in this wheat variety that confers exceptionally high levels of scab resistance, and it's detectable with a DNA marker, and that the results of that DNA marker test correspond to the very high levels of scab resistance that we see in the field. So that's kind of like this is the traditional plant breeding that we do, where we make the crosses. And there's years and years of selection, so it can take between 10 and 14 years by that route. Now, one other method that we're using, and this is a project we're funded by from the Michigan Wheat Checkoff, is to use double haploid technologies. Now, this is something that people have been using in maize breeding for years now, and that's part of the acceleration in the breeding process and variety development in maize. And we actually have a service in Kansas, Manhattan, Kansas, where we can send them F1 seeds, and a year later, they'll send us back true breeding lines, fixed lines, that we can actually take out into the field and do replicated trials with. And that cuts 5 to 6 years off the entire breeding process. So instead of taking 12 to 14 years, we're looking at 6 to 8 years before from where it goes from crossing in the greenhouse at Michigan State to seed increase and being grown on your fields. So that's one technology we're using. Another technology is genomic selection, and that's, are any of you also beef producers or dairy farmers? Okay, so they're using that same technology in herd improvement, where they can take a tissue sample and look at the DNA of that sample and determine what specific traits it will have. In dairy, you know, high feed efficiency or butter fat or milk yield, we can actually predict what kind of levels of scab resistance or yield potential that this line will have based on DNA marker evidence. So we do traditional breeding, and also what you would kind of call the next generation plant breeding, where we're DNA marker technologies. And so these are the same technologies that are being used, you know, in the private sector, large companies, and we can do these same things very well in our public breeding program.